So, good morning, everyone, and thank you for this opportunity to share the latest results from our study. And yeah, I'm looking forward to the discussions. So, working memory is the ability to temporarily maintain information about a stimulus that is no longer being perceived, and it's important for several different things such as language, reasoning, future planning. And specifically, tactile working memory has been studied extensively in both monkeys and humans using single stimulus features, for example, frequencies. Recently, in monkeys, this work has been extended to include categories of stimulus sequences, wherein the monkeys had to decide if a train of electrical pulses delivered to the median nerve was grouped, as shown on the left, or extended in time. The researchers recorded from neurons in the dorsal premotor cortices and found that the firing patterns in these regions differentiated between both the stimulus properties as well as category membership. Furthermore, in the visual domain, categories have been assessed using directions of motion, wherein monkeys had to categorize random dot motion according to an arbitrary boundary. And this has also been done with pairs of shapes, which monkeys were trained to associate together in pairs. The researchers found neuronal firing patterns in the intraparietal sulcus that differentiated between both the categories of motion and the stimulus pairs. So the current study aimed to further extend this research to humans, and 36 participants completed 4 experimental runs of a delayed match to sample paradigm, which I'll detail shortly. And the data was analyzed according to a whole brain searchlight decoding analysis with cross-validation across runs. We used 4 stimuli, which consisted of trains of 4 electrical pulses, and participants were introduced to the stimuli in a separate training session and then instructed to group the stimuli in arbitrarily defined groups. And the experimental design allowed us to have different subsets of participants with different groupings. So like I said, the experiment follows a delayed match to sample design, with each trial beginning with the presentation of 2 stimuli to the left median nerve, and a retro cue indicated which of the 2 stimuli should be encoded in working memory. The experiment consisted of 2 conditions: the stimulus condition, which wherein participants were told to encode only the cued stimulus in working memory, and the group condition, where they should additionally encode the group member in working memory. So for example, if participant was cued to remember stimulus 1, in the stimulus condition, they would encode only stimulus 1 in working memory, whereas in the group condition, they would have to remember that, for example, stimulus 1 and stimulus 2 were group members and encode both of the stimuli in working memory. Then after the working memory delay, a target was presented, and participants indicated via button response whether in the stimulus condition the target was the same as the encoded stimulus, or in the group condition if it was either the same stimulus or the group member. And then the data from the working memory delay period was modeled using sets of finite impulse response regressors for each stimulus independently and each condition. We then used these regressors for the following decoding analysis. So at every voxel in the brain, we defined a 4-voxel radius searchlight and extracted the runwise beta estimates from those voxels contained within the searchlight. Let's pretend that these boxes are the pattern vectors extracted from a searchlight for the different stimuli in the stimulus condition. We then used these patterns to train and test support vector machines' ability to differentiate between the pairs of stimuli. In this study, we used 4 different stimuli, which means that there's 6 pairs in total. This was repeated at every voxel in the brain, and then the decoding accuracy was entered as the result for the center of the searchlight, resulting in 6 whole brain decoding accuracy maps for each participant. We then averaged the 6 pairs together to create whole brain's subject-specific working memory maps. And these are the results for that decoding analysis, and we have identified a large frontal parietal bilateral network that maintains this information in working memory, including the intraparietal sulci, the premotor cortices, the inferior frontal gyri, and the supplemental motor area. You may recognize these regions as part of the task-positive network, which is active when subjects are actively trying to perform a task. And here we show that these regions additionally encode the stimulus, or in this specific context, the stimulus that is encoded in working memory. So next we explored how grouping the stimuli would affect the localization of these representations. So the groups were made by grouping pairs of stimuli. We then performed the same analysis as in the stimulus condition. However, now there's only 4 pairs. Because we're interested in where the group representations are, we don't include pairs of stimuli from within the same group, like A1 versus A2 and B1 versus B2. Then we averaged the 4 classification results together to create the group-specific working memory maps. And here are the results of that overlaid on the stimulus decoding analysis. As you can see, there's a lot of overlap between the 2 analyses, which is shown in green here. So we identified overlapping group information in the previously identified regions of the stimulus decoding analysis, as well as group-specific information in the left frontal pole and the precuneus. However, up to this point, we haven't conclusively shown that the group representations comprise anything beyond the physical properties of the stimuli. So there are still physical differences between the stimuli in the group condition. Thus, when we classify pairs of stimuli in the group condition using the previous analysis, we can't be certain that it's not just the stimulus properties driving the analysis. So we performed another classification analysis to try and disentangle the group representations from the underlying physical properties. We first trained a classifier to differentiate between 2 stimuli from the 2 different pairs of stimuli from the 2 different groups. So in this example, A1 and B1. Then we tested the classifier's ability to differentiate the remaining 2 stimuli, A2 and B2. So the nice thing about this decoding analysis is that the classifier shouldn't be able to use the physical properties of the stimuli to differentiate between the pairs, because while it could learn the physical difference between A1 and B1, that wouldn't enable the classification of A2 and B2, because the physical differences between the training and the testing pairs are different, or as I like to say, they're differently physically different. However, if the classifier learns to differentiate between the training pairs using the group representation, then it will also be able to differentiate between the testing pairs. So we repeated this analysis for all possible combinations, and I have some preliminary data to share with you today. We identified the left dorsal premotor cortex as containing group-specific working memory information that is not limited to the physical properties of the stimulus, thus replicating the findings from Rossi Poole and colleagues, which identified neuronal firing patterns in monkey dorsal premotor cortex using similar stimuli. Next, we identified a region in the left superior parietal lobule, which may be close to that reported by Fitzgerald and colleagues, who identified categories of visual motion and shape categorization in the intraparietal sulcus. Furthermore, we also find group information in the right hippocampus. The contribution of the hippocampus may have been anticipated due to the nature of the stimuli used in this task, which require the temporal binding of individual pulses into single stimuli and the single stimuli into groups. So there's an ongoing debate regarding the role of the hippocampus in working memory. However, in the context of this experiment, we believe that it may be responsible for binding the individual pulses together and the stimuli into groups, and then representing this information in working memory. Finally, we identified higher visual regions, specifically V4 and the lateral occipital cortex, as containing group-specific working memory information, and this finding even survives multiple testing correction. We believe this result is due to the strategies that participants reported using to remember the stimuli and form the groups. Around half the participants reported using visual imagery strategies wherein they formed images of the stimuli and groups, and because there shouldn't be any physical information in the visual areas, the group representation is the strongest, which is why we see such a powerful finding. So, to summarize what I've presented to you today, we identified a bilateral frontal-parietal network of regions which represent stimuli composed of sequences of medial nerve stimulation during working memory. Furthermore, when participants were instructed to remember groups of stimuli, the working memory information was represented within a similar, mostly overlapping network. However, when we controlled for stimulus properties, we identified the left dorsal premotor cortex, the left superior parietal lobule, the right hippocampus, and higher order visual regions as representing groups of stimuli, groups of tactile stimuli in a format that extends beyond the physical properties of the stimuli. And with that, I would like to thank you for your attention and all of my colleagues who helped me in this study, and yeah, thanks.
I have a question.